Hello, my name is Rick Corley. For over the past 35 plus years, my research has focused on the development of pharmacokinetic models that are used to predict the fate of chemicals as they're absorbed, distributed, metabolized, and eliminated from the body. This particular presentation is going to focus on more recent progress in development and application of three-dimensional computational fluid dynamics, otherwise known as CFD, models for the respiratory system, for comparative dosimetry across species and within vitro systems. But first, I'd like to provide a brief context of where CFD modeling fits within the repertoire of computational approaches used historically in toxicology. I go back to the beginning issue of animal studies in the past have been conducted, and then the results are scaled and extrapolated to potential human responses. Up until the early 2000s, this scaling and extrapolation involved the use of pharmacokinetic models to assist in that process, in particular the development of physiologically based pharmacokinetic models that can work to determine and predict an effective tissue dose that leads to an adverse response. More recently, the CFD-based approaches have expanded upon this approach to address the importance of 3D organ structure. Since the landmark 2007 National Academy of Sciences report, Toxicity Testing in the 21st Century, there's been progressive emphasis towards improving our ability to measure and model biological pathways in human cells, currently referred to as adverse outcome pathways or AOPs, that lead to adverse outcome rather than apical endpoints in animal studies. But note that defining the biologically relevant dose to target tissues has always been a key component for extrapolations, even with a focus on adverse outcome pathways, whether the studies are in vivo or in vitro. In 2012, National Academy of Sciences also released a report that redefined exposure science as the dose to target receptor that drives the adverse outcomes, bringing the two formerly disparate fields of research together. Pharmacokinetic modeling sits at the juncture between these two fields, and NAS reports. So today we're in a current period of transition towards achieving this vision. Efforts to reduce the reliance on animal toxicity testing, or at least be smarter about how they are used, are occurring globally. This includes the development of suites of in vitro platforms with increasing complexity and relevance using human cells, and to benchmark and validate these new platforms. Similar approaches with animal cells are needed to develop appropriate scaling relationships for in vitro to in vivo extrapolations based on target dose in all systems. For the respiratory system, the subject of this webinar, we're no longer just using in vitro systems to derive estimates of metabolic rates or other processes important to pharmacokinetics. We're beginning to use these models, including CFD-based approaches, to relate dose-response relationships from in vitro systems to real-world human exposures to materials in the air that we breathe, whether they're biological, chemical, or other origin. So in a companion webinar presented by Dr. Paul Hinderleiter for Syngenta, one such example of this comparison was presented. In their approach, Syngenta proposed a source-to-outcome analysis for a pesticide that they're undergoing current re-registration practices for the US EPA. In that approach, Syngenta chose not to conduct an additional required 90-day rat inhalation study, and instead focused on in vitro studies in human cells and enhanced characterization of potential exposures and target dose that are relevant for risk characterization. This is consistent with those two NAS reports. In the earlier studies, the primary outcome was upper airway irritation in rats. So they need to conduct a longer-term 90-day study. We already knew the outcome. It was going to be progressive chronic irritation and cytotoxicity in the nasal system that limits exposure. So instead of conducting a 90-day study where the outcome was a foregone conclusion, Syngenta proposed and conducted a series of in vitro studies using mucil at an air-liquid interface to expose and develop dose-response relationships for a variety of endpoints for cytotoxicity. In addition to that, they focused on fully characterizing the particle size and distribution of inhalable particles under each types of human potential exposure scenario that expected throughout the product chain of using the pesticide. This information is integrated with CFD-based modeling approaches that put the real-world exposure in comparison with the in vitro results to more effectively determine the ultimate human equivalent concentration and risk characterization. So what is CFD modeling, computational fluid dynamics? Well, basically, it's a numerical method for describing fluid flows. The Navier-Stokes equations form the basis for these calculations, and they describe the flow of a viscous fluid, which in this case is going to be air for the respiratory system. The solution is generally a flow velocity field over space and time, and it's this velocity field that is carrying materials throughout the airspace and over the course of the simulation. These equations, along with other supporting equations, are solved using 3D computational meshes with appropriate boundary conditions, such as the shape of the airway, the mechanical properties of the fluids, and pressures, and so forth. These methods have been widely used in aerospace, automotive, energy, and assorted physical sciences fields and engineering fields, and have been heavily used to improve the design, troubleshoot, and decrease costs in product development. In biology, though, the applications have lagged behind, but recent developments working with the heart, blood flow, fluid structure interactions have started to invoke the use of these tools, mostly available in engineering mathematical sciences. So, in essence, why so few biological applications? Well, first, it's significantly difficult to attain 3D and ultimately 4-dimensional structures where the 3D structures are now moving or changing over time, and then it is to do with a physical sciences field. And unfortunately, I hate to say it, but math phobes still exist in biology. So to do this generally requires interdisciplinary teams between the biologist and the physical scientists to develop these models. So over the past 15 or so years, respiratory and cardiovascular CFD models have become a rapidly growing area with the advent of new imaging and image analysis tools that I'll talk about later. So moving to the anatomy of the respiratory system, here I'm showing CT scans of the rat, the rabbit, and the human, starting at the tip of the nose and moving down through the upper conducting airways for the rat and rabbit, and the human. What you'll see in dark, as I'm showing here, are the actual airways. And with these new imaging techniques, here's the rat on the left, the rabbit in the middle, and the human on the right, we can now effectively construct 3D airway anatomy quite easily and readily, whereas 15 to 20 years ago, this was not possible. With this change, using imaging techniques, we are now able to develop CFD models for respiratory system much more quickly and easily. But interesting to note that there is a fundamental difference in airway anatomy across species and even between individuals. You'll see the rat nose is a highly complex structure with significant areas of turbinates in the back of the nose lined with olfactory epithelium. The rabbit also has a very complex airway anatomy. In the front of the nose, there are turbinates as well as at the back, which dictates how air flows and materials can transport into these systems. Whereas in the human, the nasal anatomy is much less complex. Furthermore, there are differences in branching patterns in the deep lung and through the larynx and the conducting airways. So all these factors are important in developing of CFD models and how air flows move throughout the respiratory system and are able to deliver agents to local sites producing their effects. So in this slide, I'm showing about a little over a decade's worth of research that allowed us to exploit the power of medical imaging technology to develop computational models, moving from improving imaging techniques to feature segmentation, ultimately creating a surface of a particular feature of interest, annotating that surface with different regions of interest or cell types, and creating these three-dimensional computational meshes that are used for all CFD simulations. More recently, 4D imaging has taken hold, and we're able to start developing multiple image sets with image registration procedures for looking at airway and tissue mechanics and the influences of disease. What once used to take several months or up to a year or more to create a single model can now be done in hours or days. And in the next few years, you'll see more and more personalized models developed. So in our laboratory, we've now developed a suite of CFD models and data sets for model performance evaluations in a variety of species that we have provided to researchers around the world. So getting back to the Syngenta proposed approach, these are simulated results for aerosol deposition under conditions of the 2-week inhalation aerosol study that produced a cytotoxicity and irritation in the nose. Here on the left shows the upper conducting airways of a rat exposed to the lowest observed adverse effect level and particle sizes used in that study. And you can see with these red dots, the location of deposition occurs in the front part of the nose near the dry squamous epithelium lining the nostril. Irritation is observed most significantly up here in this respiratory epithelium lining the front of the nose, and then back towards the larynx, being the driver for the effects that they've seen in the 2-week inhalation bioassays. Similar simulations were conducted for the human to show that there is a difference in deposition patterns. Whereas in the rat study, almost 60% of inhaled particles are expected to be deposited in the nose. Most of the inhaled particles of this side can bypass the nose and deposit along the conducting airways, down the larynx, and in the tracheal bronchial region of the respiratory system. So using that process, we ran a series of simulations with the human model using the aerosol sizes developed characterized for the different types of potential exposure scenarios. You'll see here, and this slide is actually shown in Paul Hinderleiter's webinar as well. The size of the rodent study are 2.7 microns, but humans working on mixing and loading of pesticides are at 13 microns on average, and applicator exposures are to larger particles of 35 microns. We ran a series of simulations covering a broad range of aerosol sizes, and from these simulations we can reconstruct polydispersed aerosol sizes fitting against the matched studies that Syngenta has done with worker exposures. Well, what I should point out here is rodent studies are conducted at this smaller particle size, and you can see here at 3 micron, very similar to the 2.7, where the particles are largely depositing, and most of them actually escape these upper airways and into the tracheal bronchial region of the lung. As you get larger and larger towards this particle size that are observed from the mixer loader or the applicator exposures, you'll see that particles are now starting to deposit more and more in the nose, and as they get larger, the effects of gravity drop them towards the base of the nose, into the larynx, and eventually as you get towards the largest particle size, hardly any gets past the nose and into the conducting airways of the lung. Syngenta has used these simulation results to calculate human equivalent concentrations based on the dose response analysis from the human in vitro studies, and you'll see from that webinar by Dr. Paul Hinderleiter the results from those simulations. So other potential examples, I'm going to refer to a conference recently held by the Institute for In Vitro Sciences on development of in vitro and computational approaches for studying reactive issues associated with cigarette smoke, tobacco use, and e-cigarette vapors. So in that conference workshop, so a number of research presented, including myself, on approaches for improving our ability to test cigarette smoke and e-cigarette products now that they're under regulation by FDA. So as presented in that workshop, CFD-based models have been used for reactive aldehyde constituents. In this case, these models are CFD-based, but on the surface of these airway models, we've attached PBPK models that describe the tissue. So CFD model works on airflow, and it uses to carry these aldehydes during the exposure. And the PBPK model then transfers the material based on diffusion and partitioning into the mucus, in the epithelial cell layer, and the submucosal layer. Each of these PBPK models are on every single surface facet of the CFD model and annotated by specific cell type or region, whether it's squamous epithelium at the tip of the nose of the rat, to olfactory epithelium in the back of the nose, and various regions. And we've utilized existing data to map out cell distribution in the human as well as in the monkey for simulations. So this coupled CFD PBPK models have been used to derive estimates of prior studies where rats are exposed or monkeys are exposed to cigarette smoke and compare those to human measurements during the active breathing process for exposure to cigarette smoke containing several of these reactive aldehydes. For example, I'm showing here results from simulations of an acrolein exposure in a rat inhalation study. Over the course of the inhalation cycle, as you move through inhalation phase and then exhalation. And from this, we can determine local tissue concentrations of these reactive aldehydes. In this case, we can look at how much area gets covered with the aldehyde compared to what's in the air concentration and what the tissue loading looks like. For example, here in the anterior respiratory epithelium, you can see a plot as the acrolein is building up in the respiratory epithelium during inhalation and dropping back down during the exhalation phase. And from these, we can determine internal doses that can be compared across species and to humans under realistic exposure conditions as shown in this slide. In this case, we're using a human puffing profile with oral inhalation as opposed to nasal breathing in a rat, using a measured profile and smoke aldehyde constituents for acetaldehyde, acrolein, and formaldehyde. These simulations have been conducted and then compared back to rat studies to look at the relative risk of airways in the human under smoking behaviors versus those in prior conducted animal bioassays. So understanding delivered dose isn't just critical for the in vivo comparisons, it's also critical for in vitro exposures as well. At that same workshop, the vitro cell system was presented as being used for exposing human cells to different aerosol exposures derived from cigarettes. And these are summarized in the workshop scenario, workshop summary by Bursing et al. And later, CFD modeling was used to describe the dose in the in vitro system by Lucci et al. So CFD modeling has found its way not just in the cross species, but in the in vitro and in vivo comparisons that are necessary to move forward with those National Academy of Sciences reports. Lastly, I want to throw a shout out to the National Heart, Lung, and Blood Institute Lung Map Consortium, which is now working to create an atlas of lung development where we look at where cell types and their function exist through development across the entire respiratory system. This type of data is going to become publicly accessible through the web, and at the top is the web address. And we'll also provide tissue and reagents to the medical research community. As in vitro systems are developed for the respiratory system, this type of resource will be invaluable. So in summary, the time to develop 3D CFD models has been greatly reduced over what it was 15 years ago. Imaging and high resolution analysis capabilities have been key to this. And likewise, the development of software and hardware infrastructure has also improved over the past 15 years to where now simulations can be run in a very complex scenarios such as the full respiratory system, transient breathing, and with the effects of disease. These models are now based on realistic anatomy, physiology, and the physics of airflow and material transport. This does reduce assumptions and extrapolations. And it also significantly improves the resolution in our dose response analysis. In the end, the animal use can be significantly reduced. A variety of exposure conditions can be simulated across species, and exposures can be tested in silico before actually having to conduct experiments. And that's overall significantly improves experimental design. By developing CFD models for the human, we can develop human equivalent concentrations for points of departure derived from both in vivo and in vitro studies in the future. And I should note that all models that we have developed are available, and existing templates will enhance new model applications. So as I mentioned earlier, the work to develop CFD models of biology does require an interdisciplinary team. And on this slide, I want to acknowledge my colleagues back at Pacific Northwest National Laboratory, University of Washington, University of California at San Diego, Syngenta for their contributions to the work that I described here, and the Heart, Lung, and Blood Lung Map Consortium. Thank you.